Hello and welcome to part 2 of our fungus series. In this video we'll be talking about the large group called Ascomycota. We'll briefly talk about yeasts, lichens, water molds, and slime molds. Alright, let's get to it. The learning goals of this video are to know the phylogenetic relationships between not only just the fungi, the true fungi, like we talked about in the first video, but also the slime molds, water molds, plants, and again, yourself. We're going to learn the morphology of the other type of mushroom, the cup mushroom, in this video. We'll understand how fungi and Ascomycota reproduce with ascospores. We'll understand the morphologies and symbiotic relationships found in the lichens. And then we'll also very briefly learn about yeasts, slime molds, and water molds. Here's a list of your vocabulary, just a tiny bit of overlap from the previous video. Lots of these terms are taxon names. So again, we've got some of those things that end in mycota. Those are phyla. Let's get into it. Ascomycota. This is the probably more well-known phylum out of everything we'll be talking about in this video, at least, but still, I think, slightly less well-known than the Basidiomycetes from the previous video. But Ascomycota, this is another group of true fungi. So again, only the Ascomycota and Basidiomycota are the true fungi. After I'm done talking about this little chapter of Ascomycota, I'll have to show you a cladogram as to where the other taxa are sort of related relative to the true fungi. So in Ascomycota, one of the common names for this phylum is the cup fungi, CUP, cup fungi. And we've got a couple of really well-known things, and some nice treats, like truffles and morels are found in this family. These are edible members of the family. Truffles especially are they're a very special mushroom delicacy. They're found underground, actually, and you kind of have to know exactly where to start digging to find them. And one way people do that is actually they train like pigs or even dogs to smell that scent of a truffle so that they know where to dig underground. Fun fact. Okay, so these generally have more, or members of this phylum generally have more of this sort of circular cup shape. They're usually sort of concave, and the spores are generally produced sort of right in there in the cups, and that's what we're going to talk about a little bit. Now, morels, these are a little different looking compared to the other members of the family. Just a bit different structures. But the same types of spores, same types of reproduction. So here is a summary of their life cycle. Again, there's kind of a lot more details on here than what you need to know. So let me kind of circle here what you do need to know. You need to know that their hyphae are generally haploid. This is just like Basidiomycota. But again, just like Basidiomycota, there are also some dikaryotic hyphae. These are the ones that have 2 unfused nuclei per cell. Remember, the haploid cells only had 1 nucleus per cell. Okay, well, it's these dikaryotic ones that are going to undergo karyogamy. Remember from the last video, karyogamy is the fusion of 2 nuclei. And essentially, it's kind of the second part of fertilization, right? Plasmogamy plus karyogamy really equal fertilization in the perspective of the plant life cycle and animal life cycle. They just happen in 2 very different stages in Basidiomycetes and Ascomycetes. Okay, so those 2 haploid nuclei fuse in karyogamy, and then they become a zygote, and the zygote will mature. It'll sort of mature into these 8 ascospores, okay? And so the ascus is the term for the structure that produces ascospores. So this is the young ascus, and then this, all of these, this one, and this one, these are all the maturing stages of the ascus. And at the very most matured stage, we end up with exactly 8 ascospores. Cool. Well, it's each one of those little spores that's going to germinate a new hypha that's haploid and start the life cycle over again. Very, very similar to Basidiomycota. I think I had this definition in the previous video. Actually, I think I had all of 3 of these. Plasmogamy is when the cell fuses, but the nuclei don't. Karyogamy is when the nuclei fuse. And then dikaryotic is sort of that type of cell that's in the middle, the stages in between plasmogamy and karyogamy. So there's 2 haploid nuclei. Okay. Well, an Ascocarp is the term for that reproductive cup-like structure. And here's another one. There's many species of Ascomycetes. This is just one species that sort of has that orange outline. And let's really briefly talk about the sort of life stages here. Now, I've taken one of those Ascocarps, and I've just sliced it right down the middle, and sort of we've zoomed in here, and imagine we're looking at this diagram of this microscopic look through a longitudinal section of one of these cup-like Ascocarps. Okay, so we're seeing some dikaryotic cells. Those are the cells that have 2 separate nuclei. And the plasmogamy is forming in this giant structure down here. I'm really not going to hold you to the terms, but it's really interesting that there's this one kind of, in a way, kind of a giant cell that has a bunch of unfused nuclei. And that's really where those dikaryotic hyphae are born. Okay. Right. Yeah, notice that all those dikaryotic hyphae are coming from that giant structure in Ascogonium. I'm not going to hold you to that term, but that's the structure that's sort of acquiring haploid nuclei and then allowing them to pair off. Okay, but a lot of these other hyphae are just plain old haploid, right? Even notice up here at the tops of these ones, these are all we've got a bunch of haploid hyphae as well. They're not all dikaryotic. It's just the dikaryotic ones that are going to become sexual. Okay, and so really what happens here is sexual fusion to those 2 haploid nuclei start to fuse, and this is really fertilization. Now, what this ascus looks like in real life is sort of like this. These are pictures of real life pictures of asci, and they've been dyed in this blue one up here so that we can more clearly see the ascospores inside of each ascus, but really you'd have to sort of make an outline of one of those elongate structures and then count all 8 ascospores to know that you've got an entire ascus. And again, just like in the basidiomycetes, this entire layer of spore-producing structures is called the hymenium. Cool. It's just that in Ascomycota, the hymenium layer is made up of asci, which is plural for ascus, and the asci produces 8, the ascus, I'm going to actually label this ascus, since it is just one, it has exactly 8 spores, and they're called ascospores. Now, I just wanted to compare this to the previous video. So this is now the last taxon that we talked about, Basidiomycota. They produce not an ascus, but a basidium, remember? And it looked a little different. It was one of those structures that kind of looked like this, and it had those kind of 4 filaments, each with 1 spore on the top. And then, so like this whole structure is a basidium, and then one of those is a basidiospore. Remember that? Oops. Okay, well, in a basidium, they produce only exactly 4 spores, as opposed to the ascus, which produced 8 spores. So that's one difference between an ascus and a basidium. Another, one of the similarities, though, is that they both produce these haploid spores via meiosis. But in the ascus, there's another thing that happens. It's meiosis and then mitosis. So let's go over that really quickly. Because remember, in meiosis, the result is always exactly 4 haploid cells. So if that was it, then the mature ascus would only have 4 spores, but it has more. So, okay, so let's back up here a little bit. That whole layer of all of those different kind of stages of development, all of those many different asci, that's called the hymenium layer. And what happens is first there's sexual fusion. So there's sort of going to be stages that are shown here. The first stage is those 2 haploid nuclei in a dikaryotic cell will fuse. This is also known as karyogamy, and it's sort of more well known as fertilization, really. Although karyogamy is more specific for what's happening here since it's just the nucleus. The result, as we always know, the result is a diploid zygote, which is a single-celled organism, and then that zygote immediately undergoes meiosis. The result are those 4 haploid cells, but then those 4 haploid cells each undergo mitosis. So in other words, they each double, and that's how we end up with 8 ascospores inside of a mature ascus. Cool. Yeah, we already had this. Just a little reminder. Just like Basidiomycota, Ascomycota is haplontic, meaning it's really those haploid cells that are multicellular. The diploid cell is never multicellular. It's just that one sort of moment in time where it's a single cellular zygote. Only diploid thing. Okay. Now, their Ascocarp is that whole cup-like structure. Like I circled like one of these earlier. There's an Ascocarp. There's another Ascocarp there. So we've got a whole bunch of them down here. Well, an Ascocarp is just any structure that has asci, okay? And a lot of times they are in this cup-like structure, the ones that we've been talking about this whole video so far. And here's a diagrammatic view of one of those. It's a cup. It's open to the entire world, and the spores are just released into the environment with wind or rain splashes or whatever happens to push them around. Okay, there are actually 2 other types of ascocarps, or in other words, there are 2 other types of structures that produce asci that would be found just in different species. And I just wanted you to know that there are these other 2 types. Notice that Cleistothecium and Parathecium, those are not terms on your terms list. I just wanted to show you them because they are interesting and they're an important part of this phylum. So sometimes the spores are not as wide open to the environment. Sometimes they're literally kind of inside of a structure, and that structure needs to be either broken open or somehow those spores need to be released from there. So have you ever seen this thing growing around? You could find this, I'm sure, if you walk around the community forest long enough, up in Humboldt, you will find this eventually. This is called dead man's fingers. It's in the genus Xylaria. And it looks, it's this kind of creepy looking fungus. And what happens is it's actually an ascomycete. And all of those little kind of white structures, all that white stuff on the end of these sort of black stalks, you can kind of see in the background there, all those white tips are attached to these black stalks. Well, it's the white structures. That's actually the spores. So here the asci are just produced, like, it's some crazy thing. The asci are produced inside of these structures that are lining that entire sort of finger-like structure. Okay, so let me go back. Imagine I take a cross-section through this right here. Take my fine razor and I cut it right there, and then I take another slice right next to it. And I've taken just that little tiny piece in the middle, and I've given it some nice dyes. I've put it on a microscope slide. So this thing is very small. It's only like, maybe a millimeter or 2 in diameter. That's what we're looking at here. That's a cross section through one of those. Okay. And so now if we were to look at, one of these kind of thingies, we zoom in like on this whole row of them. This is what we see when we look up close. We see these actually enclosed structures, each of which have these linear asci. Okay. So now we've zoomed in even more here, and we've got, again, one of those sort of stalks, one of those linear structures that has 8 mature ascospores inside of it, and there's just a whole bunch of them inside each one of these parathecia. So we just have an incredible amount of spores being produced on those dead man's fingers, just in a slightly different way than those cup fungi. Lots of diversity in this family, including that morphological diversity that I just briefly talked about. It's very cool. So a little recap again. I've already kind of showed you this slide briefly on the previous video. There are 5 phyla of true fungi, and then these 2 informal groups sort of within the true fungi. Now, I call these informal and all mycologists tend to call these informal because they're actually only entirely known to be asexual. This is wild, right? This is crazy. They're eukaryotes, but they're entirely asexual. They don't go through the fertilization and meiotic process like all of the other organisms we talked about this semester. And so it's sort of hard to figure out exactly where they fit in the tree of life. But they do know that both the Deuteromycetes and the yeasts had a sexually reproducing ancestor who was probably an Ascomycete. So really briefly, the Deuteromycota are really important because this is sort of where a lot of mold comes from, and things like penicillium. This is a genus that produces antibiotics and brie cheese, and also some other really delicious cheeses, actually. They come from fungi, okay? And if that sounds weird or gross, don't let it get to your head. You've been eating it your whole life, or maybe not. Maybe you don't like cheese. That's totally fine, too. Maybe you've had penicillin before, though, or maybe you're allergic to penicillin. Lots of different possibilities with this group of organisms. All I want you to know is that the Deuteromycota are asexual, and they are this informal group of fungi because they seem to be closely related to the Ascomycota, but they just don't really, they just don't evolve the same way since they don't undergo sexual reproduction, okay? Now, it's very confusing because there are hundreds of species of these things, even though they're asexual. It's a group of organisms that definitely needs study. Like, actually all of the fungi. So yeah, you know this stuff. Moldy bread can contain actually dozens of species of fungi, and some of them will be the Deuteromycetes. Some of them will be in these other groups of fungi that we actually don't have a chance to talk about this semester. And then others will be Ascomycetes. Okay, yeasts. This is another group of asexual organisms that's probably nestled within the Ascomycetes. This is what geneticists think. And what yeasts do is, instead of sexually reproducing, they do this thing where they bud. They'll just take their whole body, and they'll just sort of start to kind of imagine this hair kind of growing over time, and then it just kind of keeps growing, and then these two will split apart from one another. And now you've got two of the exact same cell. Okay, cool. This is how they spread. This is how they grow. This is how their populations grow, I should say. Yeast is very ethnobotanically important because, of course, yeast is responsible for making bread rise and making all alcoholic beverages virtually. It's going to be what turns sugar into alcohol. And this is very important for many reasons. Both socially, and medically, health-wise, alcohol is also important. And, rubbing alcohol and stuff. But bread, can't deny that. Cheese. All right, moving on. Another super awesome group of organisms we need to talk about are the lichens. The lichens are very special because it's not just one organism. It's actually two partners, two symbiotic partners. Now, there is a mycobiont, bio meaning sort of life, myco meaning mushroom. And so there's a part of a lichen that is a fungal symbiont, usually a member of the Ascomycota. And then the other member is a photobiont. This is usually a chlorophyte, remember, in algae, or a cyanobacterium. So it's going to be something that's photosynthetic. So these two live very intimately together, completely. You can't remove them from one another, and that's what a lichen is. Now, when we look at lichens, you'll see three morphological types. Now, a lot of times people will kind of sort of like see this on a branch or something and think, oh, there's some moss. Well, sometimes there is moss mixed in, but actually we have multiple species of lichen just in this one picture here. There are three major morphological types of lichens. You generally call the body of a lichen a thallus. Remember, just like we called the body of like a gametophyte a thallus, or of a liverwort gametophyte was a thallus. It just sort of usually means like a flat, broad structure of cells. So the three main morphological types are crustose, fruticose, and foliose. You kind of see the difference between them in this picture. Crustose, it's like a crust. It's something that's going to be hard pressed to either a branch or to a rock. Foliose, well, it kind of sounds like foliage. And yeah, foliose is sort of leaf-like. It's going to be something that's flat and broad. And then fruticose, these are going to be those lichens that are more branchy, sort of look like more of a network of more cylindrical filaments. Here's some more examples of those three. So some more looks of foliose, and then we have an incredible amount of diversity in lichens. In fact, to drop some more ideas for you, there's a class called Lichens and Bryophytes. We are so lucky to have here at HSU another upper-level botany class that I highly recommend taking if you ever get a chance to do so. This is, again, just really scratching the surface of what you would talk about for an entire semester in that class. Okay, so the foliose lichens, these are really kind of broad and flat. The crustose lichens, can you kind of tell these pictures were taken of lichens on a rock? So you could like scrape it off with something metal maybe if you really tried, but don't harm them. They're fine there. Just leave them alone. But like you can't like pick these off. These are really hard to press to the rock or to the piece of wood. Fruticose, these are the more branching ones. They come in a lot of different looks. Sometimes they have these cool sort of puckered lip looking structures at the top. Other times it looks kind of like mossy hair hanging down. This is not moss. This is a fancy type of lichen. And sometimes, yeah, you can see them dangling in copious amounts from things like spruce trees on the dunes and all sorts of other places. Very cool. Very cool stuff. What's so cool about a lichen is that, like I said, it's two organisms in one. So down here we have sort of a cartoon diagram. And then in this picture labeled B, we have an actual look of a very tiny section of a lichen thallus. And by actual, I mean like this is the actual colors. There are these little green specks. Yeah, those are the photosynthetic organisms that are surrounded by the rest of this stuff, which is fungal tissue. And then in this picture is just more of a look at one of those, more of a detailed look at the same thing as was in B. It's just that in C we've given them some dye so that the different organisms can kind of stand out, increase the contrast. Now, the lichen thallus is built by the mycobiont. This is the fungal symbiont, and that's going to make up the majority of the mass here. It's what's called the upper cortex and the lower cortex, and then mixed in is the photobiont, okay? The photobiont is that photosynthetic organism. So usually lichens are named and classified based on their mycobiont while keeping in mind the photobiont. So we tend to call species of Ascomyces, we call them lichenized fungi, if this is their form, if this is their main form. And remember, a mycobiont, this is usually an Ascomycete, could be a bit of a Basidiomycete though. And the photobiont, usually a green alga, a chlorophyte, could be a cyanobacterium too, though. The way that they reproduce is, well, both sexually and asexually. You don't need to know this word. I just want you to know that fungal spores will just have, algal spores will have these fungal hyphae just wrapped all around them. That's what you're seeing in this picture. This is an algal spore or maybe a cyanobacterial cell with these fungal hyphae all around. So when they reproduce, they sort of spread together. Or they'll just, like a piece of their thallus will break off and they'll continue growing. But really, in a way, they resemble Ascomycetes. Check it out. There's little Ascocarps everywhere. Remember those cup-like structures? Ascocarps. Here's more. They look just like the cup fungi. It's because they are. Cool. It's just that they're photosynthetic. I know. Now, even this like orange and yellow one, it might not be green and photosynthetic, but it's got a photosynthetic layer in there. It just also might be producing some other sort of pigment. Okay, whoa, moving on. We're done with the lichens now. Totally moving on here. Do you remember the Heterocons? Remember the Heterocons way back in the day? We talked about brown algae. And we talked about diatoms, both of those really kind of briefly in week like 11 or something, or 10. And we talked about them in light of algae. So why am I bringing that back up right now? And what did a heterocont even mean? Well, there's this organism. Yeah, remember Bacillariophyta and Phaeophyta? Well, there's this organism that we actually biologists used to think was a true fungus, but then they realized it's actually more closely related to the brown algae and the diatoms than it is to Basidiomycota and Ascomycota. And that's this thing called Ooomycota. So Ooomycota is the phylum, and it is actually within this group of organisms called the Heterocons, which, remember, the Heterocons, it's just this group that has diatoms, algae, and lots of other things, actually, that we don't talk about in this class. What they have in common is this heterocont flagellum at some point in their life cycle. Hetero meaning other. And what they have is two different types of flagella. At least one stage in their life cycle. They have two different types of flagella. And that's at least one of those morphological features that is shared between them all. And it's evidence that they're closely related. That Ooomycota is more closely related to brown algae than it is to an ascomycete. Okay, so water mold, that's the common name for this phylum. These are non-photosynthetic, unlike the diatoms and the brown algae. And they are filamentous, so just like the true fungi. And this is what fooled early biologists into thinking that this was a true fungus. So Ooomycota, you definitely do not need to know any of these life cycle terms whatsoever. I just need you to sort of be able to recognize Ooomycota with that common name. And I want you to know, I just kind of wanted you to have this if you were interested. This is more important, their relationship with humans. Ooomycota can be a very aggressive plant pathogen. Not generally a mammalian pathogen, actually. Sometimes they'll infect insects. But the problem is that they will attack like potatoes or oaks. And these are different species of members of this phylum that will completely wipe out giant populations, and it has caused human suffering in light of starvation, famine, by causing the potato late blight. So Phytophthora ramorum, this is a more locally well-known pathogen that affects oaks in our area, and a lot of oaks. It's really bad. Some people are doing research on this actually at our school, so look into that if you're interested. Ooomycetes have been around for a really long time. They're really noticeable because they have this globular structure that sort of produces these egg-like structures inside. And that's actually where the term Ooomycota comes from. It's like egg, it kind of stands for egg, and then mycota, fungus, like an egg fungus. And that really distinctive look has been found in fossils from up to 410 million years ago. Okay, so let me show you about where they would be sort of located relative to the Ascomycetes and the Basidiomycetes. So over here, we've got, let's say, Ascomycota and Basidiomycota. Now you are actually more closely related to these organisms than anything else that we're going to be talking about. Okay, now the Heterocons are way over here. We have Bacillariophyta and Phaeophyta, the diatoms and the brown algae, respectively. They're more closely related to each other than either one is to the Ooomycetes, but the Ooomycetes are very closely related to them. Now, the truth is that the photosynthetic algae we talked about, Rhodophyta, and the true plants, all of the things with true green chloroplasts, the Viridiplantae, these are more closely related to each other than either are to this group of Heterocons. Okay. And now we only have one more group that I need to add into this before I can connect these two. And it's the one that I'm going to be talking about next. It's the slime molds. And this is going to be the last, actually, organism that we talk about in general botany together. It's Myxomycota. Let me write that in. That'll be on the next few slides, like I said. These are the slime molds. And they're more closely related to the fungi and the animals than anything else over here. So this is the running hypothesis for the relationships between really all of the organisms that we have ever talked about in this class. Okay, and the ones that we're talking about today are all over the place, really. We've got the oomycetes over here, more closely related to the other plants. And then we've got all of these other true fungi and then fungus-like things over here. Okay, so this is showing the relationships. Now let's talk about this very last group. The slime molds, I kind of wanted to save them for the end. They are so unique in so many ways. And their evolutionary relationships has been sort of very confusing for systemicists for a very long time. They weren't sure if it was a fungus, if it was an animal, because here's the thing. Slime molds can move. What? Okay. So again, phylum Myxomycota. This is the phylum name for the slime molds, or maybe more well known as the plasmodial slime molds. Now, these structures oftentimes are not moving. You're not going to see them move with your naked eye unless you're sitting in the same place for a very long period of time. They can cross the street or essentially make their way up a branch or across a log or something like that over a few hours. It really doesn't take that long. And they're moving by sort of pumping this fluid around within this one body. This is wild. What they have is a giant body, but they're not made up of cells, okay? They're not made up of individual cells with individual nuclei. Instead, it's just one mass of a bunch of stuff, one without cell divisions and a bunch of different nuclei. And every once in a while, they produce these reproductive structures, as we're seeing in some of these pictures. So this is their stage when they're moving around and feeding. It's called a plasmodium. And then the other stage where they're reproducing and producing spores is the sporangium stage. Now, like I said, they can literally move around. And it can actually learn. It's this kind of organism that people have done some research because it can kind of learn. It can figure out how to get from point A to point B quickly. And what's happened in this experiment here is they just started off by having slime molds. This organism just kind of threw out this maze, and then they put a piece of nutrient here and a piece of nutrient here. And it very rapidly sort of found the connection between the two, and then notice it eliminated all of the other kind of excess, all those other excess hyphae that were no longer needed. And so now this is the most efficient route from one nutrient to the other, and this is sort of where they'll put all of their entire body is right on those nutrients. To do this very rapidly. You can definitely look up some cool YouTube videos of slime mold time lapse. Yeah, very cool stuff. And even cooler are their reproductive structures. Now, sometimes they are truly called sporangia, but other times they're so fancy that they have other fancy names, and you might learn about those in some other potentially upper level botany classes, or if you are interested in them on your own, there's lots of good resources out there for learning more about slime molds. Really intricate reproductive structures sometimes. I took some of these pictures, these two, just last summer. And so you can definitely find a whole lot of different species in our area. And they fruit all really kind of throughout the year. Different species will sort of present themselves in different times. And it's really cool to see both styles, both the plasmodium and the reproductive sort of version of these. I was so fortunate one year, this was a couple of years ago, to wake up on Christmas morning, actually, to this large structure on the inside of my front door. And I was so confused about it. It definitely looked like a fungus or something. I had no idea. Well, a couple of days later, you can see what happened here. This was sort of day 1. This was, I believe, day 4 or 5. And it just completely hardened. And then by the next day, that outer structure fell off, and then I took a knife to it, and I was just so curious. It's just a mass of these spores. And I found out, doing a little bit more research, this was a very fancy type of slime mold that I was so lucky that it presented itself in its reproductive form inside of my home, right next to my door handle, as you can see right there, on Christmas a few years back. So, very special. No idea why I chose my door. This is just in case you want to know about the life cycle. I'm definitely not going to talk about the details. I just want you to know about this part here, the multinucleate plasmodium. This is what I was talking about earlier. It's just this giant structure that's not divided into multiple cells. it's just feeding all like this one mass. There's nothing else like it in the animal kingdom or the fungal kingdom or the plant kingdom at all. It's wild. And you can find these here in Arcata, if that's where you're at. The dog vomit slime mold is around, especially this time of year right now. You might find it in either its plasmodium or its sort of fruiting stage. Very exciting stuff. And I hope you have enjoyed this, and please feel free to come by my office hours this week or next if you wanted to talk about any of these organisms in a bit more detail. All right, see ya.